students, this is Professor C.P.S. Verma from Laureate Institute of Pharmacy. Now, today we will discuss the next chapter of your syllabus, that is glycosides. What are glycosides? We will discuss. These are organic molecules plus sugar moiety, mostly monosaccharides, and definitions. Then organic compounds from plants and animal source which on enzymatic or acid hydrolysis give one or more sugar moiety glycon with non-sugar moiety aglycon genin. So in glycoside there is two parts, one is glycon and aglycon, but on acid hydrolysis it will dissociate into aglycon moiety and glycon moiety, and aglycon moiety is also called as genin. Then acetyls or sugar ethers, it is also called like this. Then acetyls or sugar ethers, then found by hydroxyl group of each of non-sugar and sugar moiety with loss of water. OH group of aglycon may be alcoholic or phenolic, and in some cases amines also there. Then sugar involved in glycosides are of different types but mostly beta D glucose. Then classification of glycoside, either of chemical nature of aglycon part, therapeutic activity of aglycon part, linkage between glycon and aglycon. So physicochemical properties of glycosides, that is colorless, solid, amorphous, non-volatile, that is flavonoid, yellow, anthraquinones, red or orange. Give positive reaction with mollusk and Pelling solution. Test after hydrolysis. They are water soluble compounds and soluble in organic solvent. Most of them have bitter taste except populin, glycyrrhizin and stereoside. These are stereosides are bitter glycosides, and odorless except saponins, that is saponin glycoside comes under this glycyrrhizin is the saponin glycoside. When a glycoside has a lot of sugar in the solubility in water, decreases. When a glycoside has a lot of sugars, its solubility in water decreases. Then glycosides hydrolyze by using mineral acids and temperature or by using enzymes. Glycosides are water-soluble compounds and insoluble in the organic solvents. The next point is glycon part, water-soluble, insoluble in the organic solvent, and aglycon part, water-soluble and soluble in organic solvent. Then some glycosides are soluble in alcohol. The role of glycosides in the plant, converting toxic materials to non or less toxic. Then transfer water insoluble substances by using monosaccharides. Then source of energy, sugar reservoirs. Then storing harmful products such as phenol. The regulation for certain functions, for like growth. Some have beautiful colors, pollination process. Then some glycosides have antibacterial activity, so they protect the plants from bacteria and disease. Then this is the structure where some glycosides have antibacterial activity, so that protect the plant from bacteria and diseases. This is amygdalin. This is the bitter from we are getting this glycoside amygdalin from bitter almonds, and this enzymatic reaction with hydrolyze, and this will give SCN, which is antibacterial. Then classification of glycosides. See glycosides when sugar moiety is linked with carbon atom. For example, cascarosides or cascara, aloin of aloe, and carminic acid of cochineal, then mainly present in Liliaceae family, not hydrolyzed by heating with dilute acid or alkali, but by oxidation hydrolysis with ferric chloride. Then glycon is linked with the OH group plus SC. Then glycon, it will split with the glycon plus A glycon plus water. That is how the hydrolysis will take place, how it is showing the dissociation. Then O-glycosides. O-glycoside, when sugar moiety is linked with the oxygen atom, hydrolyzed by treatment with the acid or alkali to a glycon and sugar. Then like glucuronidine, glucuronidine. Then this is the example where glucon and A-glycan part will be displayed by hydrolysis. Then S-glycoside, when sugar moiety is linked with the sulfur atom. Then N-glycosides, when the sugar moiety is linked with the nitrogen atom. And some example, nucleosides where amino group of base reacts with the OH group of the ribose, deoxyribose, ultimately gives N-glycosidic form. So this is how the hydrolysis will take place. This is it shows. Then classification of glycoside based on the glycan part. That is glucose, senosides, rhamnose, preguanine, then digitoxose and digoxin, then glucose and rhamnose, glucurahamanoside and glucofrangulin, and then rhamnose and glucose, rhamnoglucoside and routine. In the routine, these rhamnoglucosides are available. Then glycoside, classification of glycoside based on the chemical group. Saponin, which the glycosides which will form the formation of foam or soap-like activity. Then A-glycan translinked with steroid like ginseng, diascoria. It is translinked with the steroid part, A-glycan part, with the sugar moiety. Then cardiac glycoside poisons from skill and digitalis. Then anthracene derivative, these are purgative in nature, and in this category comes aloe, senna, cascara. Then flavonoidal mainly anti-inflammatory drug, ginkgo biloba and buckwheat. And the next category, coumarins, cyclooxygenase inhibitor, for example, AMI and Vishnaga. These are the coumarin cyclooxygenase inhibitors. Then phenolic analgesic like aspirin. Then cyanogenic compounds, bitter almond, wild cherry, bark. These glycosides we will get cyanogenic from bitter almonds and wild cherry bark. Then classification of glycoside based on the therapeutic effects. Then CHF, congestive heart failure and cardiac muscle stimulator. That in this category comes digitalis glycosides, digoxin, digitoxin, getoxin, and this is comes from the foxglove leaves. Then cubane, strophanthus gratus seeds. Cubane we will get from the strophanthus gratus seeds. Then K. strophanthine we will get from the strophanthus combe seeds. Then cellerin A and B, which isolated from red and white skill bulbs. Then next category, depending upon the therapeutic activity, these are the cardiac glycosides which we discussed now. And next category, laxative group of glycoside, that is senoside A, B, C and D, senna leaves and fruits. Then cascarosides A and B, cascara bark. Then aloin and barberine, These are aloin and barbaloin. These are we will get from aloe vera and aloe. Barbadensis juice we will get. These are the all the glycosides we discussed. These are laxatives, which is given in the case of constipation. The local irritant group, sinigrin, black mustard seed, brassica nigra. Brassica nigra will we will get the sinigrin, यानी सरसों जिसे कहते हैं, सरसों से ब्लैक मस्टर्ड, उससे हमें क्या मिलता है, sinigrin, and that is a local irritant group. Then analgesic and antibiotic category, then salicylic acid will get from willow or salix bark. Then anti-inflammatory group, aloin for acne and glycyrrhizin for peptic ulcer. Glycyrrhizin from peptic ulcer we are getting glycyrrhizin from Glycyrrhiza glabra, यानी मुलट्ठी, मुलट्ठी से ग्लिसरीजिन निकलता है जो कि पेप्टिक अल्सर में दिया जाता है. Then class or type and example, these are different types of glycosides. Phenyl glycoside, all these category, berberine and bearberry. From the example is arbutin, and then glucovanillin, it will get from vanilla species. Then anthraquinone glycosides we will get from senosides, that is from senna, leaves or pods. Then flavones, flavones we will get from hesperidine, yani orange and lemon we will get hesperidine, and hesperidine is a flavone type of glycoside. Then cyanogenic glycoside, Vishnaga, Vishnagin, Me, Vishnaga will get cyanogenetic this glycosides. Then glucoalkaloids, that is solasudine, solanum species we will get these glucoalkaloids. Then steroid glycoside, that is cardiac glycoside, it comes under steroidal because the cardiac glycosides have the steroidal structure. So digitoxygenin from digitalis we will have the steroidal structure, so it comes under steroidal glycoside, that is cardiac glycoside. Then saponins, discorin, discorin from discoria, and coumarin from umbelliferon from asphatica will get coumarin. These are the another classification based upon the chemical compounds. Then anthraquinone, the chemical test is Braun-Taggar's test and modified Born-Taggar's test. Then flavone, there is Shinoda test. Then Grignard test for cyanogenic, and for steroid cardiac glycosides we will test by legal test and Baljet test and Killer-Killarney's test. We'll get for, we will have these tests for the cardiac glycoside. Then saponin glycoside, this foam test and hemolytic test. Then coumarin UV light test for coumarin glycosides. These are clinical tests also. Then examples of glycosides, effects, use and application. Then senosides, aloin for laxative, and digitoxin sterphanthin as a cardiac tonic, sinigrin, black mustard, GIT irritant. Then salicin will use for anti-rheumatic properties, and glycyrrhizin, glycyrrhetinic acid, anti-ulcer, and hesperidin, capillary fragility. Then methyl salicylate, Gaultheria, this use is analgesic. Then cardiac glycosides for sterols. Now purely we will discuss glycosides. What are cardiac glycosides? So, and in our your syllabus, cardiac glycosides are there because the drug digitalis we will discuss under this, which is also mentioned in your syllabus. So we will give the introduction. The Genins, a glycan part of all cardiac glycosides, are steroidal in nature that acts as cardiotonic agent. They are characterized by their highly specific action, cardiac muscle, increasing tone, excitability, and contractility of this muscle, cardiac muscle, thus allowing the weakened heart to function more efficiently. Any inotropic properties are there when we will increase the strength and it will contractility of the cardiac muscle. These glycosides are very important and given in CHF, yani cardiac congestive heart failure. This will increase the contractility and the heart will start pumping. Then this is the structure. Because you see the structure, cyclopentophenethrin ring, and this is a steroidal molecule. You see the numbering, and the electron ring is attached to the this position 13 and 14, and all this and the sugar moiety at the position sugar glycosidic linkage at position 3. Then you see the numbering, you should remember the numbering, and then R is left if it is cardinolide or buphenolide. There are two types of cardiac glycoside, cardinolide and buphenolide. Then all cardioactive glycosides are characterized by the following structural features: the presence of beta hydroxyl. You see the specific features that in cardiac glycoside it is a must that the beta hydroxyl group will be present at C3 position, because that time if the hydroxyl group beta hydroxyl group is not there at C3 position, then it will lose the cardiac activity, then it will not act as a cardiac tonic. So which is always involved in the glycosidic linkage to mono, di, tri or tetrasaccharides. So this is the structure where at the position 3 the sugar moiety is attached. The presence of another beta hydroxyl group at C14 position. C14 position another hydroxyl group is there. This is a must. These are the characteristics, special characteristic of the cardiac glycoside. The presence of unsaturated 5 or 6 membered electron ring at position C17. 5-membered ring, if it is 5-member ring, then it is cardinolide. Then if it is 6-membered ring, it will be coming under the category of buphenolides. So two types of cardiac glycosides are there. And electron ring, 5 or 6 membered electron ring at position C17, also in beta configuration. Then you see fourth point, A and B ring is usually cis, and while the B and C junction is always trans. So, and the C and D ring junction is in all cases cis and trans. Cis is closed and trans is away, like that. Then additional OH group may be present at C5, C16 and C5, C11 and C16 position. Cardiac glycosides that alpha beta unsaturated 5-membered electron ring in position C16 are known as cardinolide. Yes, just now I discussed if the ring attached electron ring is 5-membered, then it is cardinolide. These are represented by the digitalis and strophanthus group and shows slowing and strengthening effect on failing heart. So this 5-membered this electron ring is important, and it is attached at the position C17. Digitalis glycosides contain angular methyl group at C10, while strophanthus glycosides are characterized by presence of either an aldehyde CHO or primary alcoholic CH2OH group at C10 position. So you see the difference. This is in the cardinolide R, there is 5-membered electron ring. Then in biphenolide it is 6-membered electron ring. You can see the difference. It's same. There is attachment of this electron ring at the position 17, and but if it is 5-membered, it is cardinalite. If it is 6-membered ring, it will be biphenolite. So cardinalite and biphenolites, these are the two cardiac moieties, cardiac glycoside moieties, which is very important for when it is given to the this failing heart. So cardiac agents that have doubly unsaturated 6-membered electron ring in position 16 are referred as biphenolites, and this group includes the sequel glycoside and the toad venom bufotoxin. So bufotoxin derived from toad. Genus toad is the frog. There's a big frog that comes in the toad category. That is a very big frog. That is toad, and its venom is very toxic. So that bufenotoxins are collected from the toad venom, before derived from toad genus, and denotes double, and while olide denotes electron ring. So you see, diene, adiene denotes double bond, while olide denotes electron ring. Then these are they are found in higher plants. They are abundant in family Apocynaceae, Asclepidaceae, and Ranunculaceae, Sterculaceae, Leguminosae, etc. They are also found in monocots like Liliaceae family, for example, squid. They are also found in frogs, African toads. These two glycosides are distinguished according to the presence of 5 or 6-membered electron ring. We have already discussed. Those with 5-membered electron ring are cardinalites, for example, digitoxygenin, digoxygenin, and gitaloxygenin, and gitoxygenin, stofenthidine. These are the 5-membered, these are cardinalites. And then those with 6-membered electron ring, biphenodialides, are silerin A and silerin B. This group will come under the biphenodialides category. Silerin A is the cardiac glycoside, and maximum plants contain cardinalides. So cardinalides are more important, and therapeutically and practically it is given for the heart failure. But another category, silerin A, is also coming in the category of cardiac glycosides or a cardiac tonic. So modern use to treat congestive heart failure, dropsy, for treatment of atrial fibrillation and flutter, a glycan structure important for activity. The general properties of cardiac glycoside. These are the general properties. It should be amorphous powder, bitter taste, solubility in water, insolubility in organic solvent, very toxic compounds, and odorless. Then physical and chemical properties of cardiac glycoside. Solubility. Already we discussed solubility and stability. Already you see the solubility in acid hydrolysis, split sugar and from the aglycan parts. Enzymatic hydrolysis, you see there are three types of hydrolysis: acid hydrolysis, enzymatic hydrolysis, and hydrolysis at elevated temperature. So classification of cardiac glycoside according to their therapeutic effects: congestive cardiac heart failure and cardiac muscle stimulators such as digitalis glycosides. And under this already we discussed digoxin, digitoxin, detoxin, and these are cubane also from Strophanthus greatus seeds, K. strophanthine from Strophanthus combe seeds, silerin A and B, which is isolated from red and white squill bulbs, because silerin A and B coming as a biphenodienolides, and convolocides, convallaria majalis, lily of the lily of the valley. Then according to the type of the electron ring, adenylide one double bond electron ring has 5-membered electron ring unsaturated attached at its position. So this is again the repetition. We have discussed it. Cardinolides and biphenolides is chemistry, and at what there is attachment at 17 position. It is the repetition already we discussed. Then nomenclature also we have discussed, and then these cardiac glycosides occur mainly in plants from which the names have derived. Digitalis purpurea, Digitalis lanata, Strophanthus combe are major sources of cardiac glycoside. A-glycan moiety, the term dennin at the end refers to only A-glycan portion without sugar. Also we discussed this A-glycan and glycan parts. And cyclopentophenanthrene ring, already I told you that this is the ring which this structure of cyclopentophenanthrene ring is the structure of steroid. So these cardiac glycosides come under the category of steroidal cardiac glycosides. So sugar moiety also in the beginning of this chapter we have discussed what type of sugar hexoses are attached, rhamnol, digitoxose. Digitoxose, D-digitoxose is the because of the attachment of this group. This Killer-Killarney test is positive because of the digitoxose sugar attached to the A-glycan part. These are the structure of different cardiac glycosides. Then chemical identification we have already discussed, but this is the complete test, Lieberman test and Killer-Killarney test. How we'll take, how much we will take, and boil, and all this complete deoxy sugars. Dextrose, I told deoxydose. This deoxy sugars are attached. That is why the Killer-Killarney test is positive in this cardiac glycosides. Digitoxose is the deoxy sugar which will give positive test for Killer-Killarney. So take test powder drug. This is very important test. Mostly the examiners will ask what is Killer-Killarney test, and or in the MCQ it may be asked Killer-Killarney test is meant for what. So this is for the testing of cardiac glycoside, and why it will give red color because of the presence of digitoxose. D-digitoxose is the sugar, deoxy sugar, which will give the this test is positive because of this. Test for 5-membered electron ring, Legals test, then Bulger test, then Kiddy's test, and Raymond test. These are different tests. Genthidrol test, deoxy sugar. Genthidrol test will there. Then Salkovsky test is also there for cardiac glycoside. Then tolerance test, antimony trichloride test. These tests you have to remember. And then, of course, the family we have to discuss Scrofularaceae. Scrofularaceae is the family, and already we discussed that source, biological source and botanical source, Digitalis purpurea, Digitalis lanata. These families come, these two species comes under the family Scrofularaceae. Then Apocynaceae, Stephanthus will come under this category, and then skill will come from the family Liliaceae. So again, the repetition, we will discuss the digitalis. These are the details. What is this the source, and digitalis purpurea family, Scrofularaceae, then synonym, purple foxgloves, ladies gloves, finger flower. This then What is the difference? These are the different genin parts, aglycan parts, which we have discussed. Then chemical test already we discussed, Pellet-Pelani test, Raymond reaction, Deagle test, Golgi's test. Already this is the repetition of this all. Then what is the use? Already we told myocardial contraction, it will increase the inotropic property of the heart, and then it will start pumping the heart failure in the case of congestive heart failure. Then alternate and these substitutes are also there. Then this some categories are there which will be mixed with the cardiac glycide. Then will be given as such. So